a fantastic lady that is super excited about microbiology. I don't know, she might be on par with my level of excitement of microbiology. You guys know how much I love microbiology, so we are two of the same mind. So she's an integrative biologist, and she's interested in actually how the host diet and physiology on the genome and evolution are going to actually influence the host microbiome. And so her lab looks at diet and antibiotic perturbation and looking at the dung beetle's digestive system. And so her talk today is going to be focusing on looking at the microbiome of that dung beetle. She is someone who was a postdoctoral fellow at the Institutes for Genome Sciences. And she is very passionate about science communication. She actually has a great blog that you guys should definitely look into. It's called Mostly Microbes. And so if you want to read more about what's going on in the microbial world, Mostly Microbes is her blog. And so I encourage you to get out and read that. And she is really interested in looking at the human microbiome as well, and focuses a lot of her blog on the early childhood development and human microbiome's interaction. So if you want to read more about that, you can do that on Mostly Microbes. So with that, I want to welcome Dr. Ann Estes. So.
Thanks so much. Hi, thank you, Dr. Kara, for that very nice introduction. And thanks for coming out today. I really appreciate it. So, yeah, one thing with the blog, though, it hasn't been updated since I've started as assistant professor for some crazy reason. I don't know. So, I guess the last time I updated was in June, but I'm going to get back to it hopefully in the summer. So, today I'm going to talk to you, though, and I especially appreciate you coming out at lunchtime to talk, right, we're over here. Shut my computer down because I'm habituated. About dung beetles and their microbiome. And so I appreciate you coming out at lunch because I'm going to show you a whole bunch of poop pictures because it's one of those parts of life, right? I mean, everyone poops, everything poops. There's kids' books dedicated to poop. And so it's really abundant. It's all over our environment. And if you do crazy things like I do, and start foraging around, or not foraging, I don't eat out of dung pats, start looking through cow pats, you see that even one simple dung pat is an ecosystem unto itself. It's really neat. There's different layers and there's different places where you'll find totally different organisms. And I'm just talking about macroorganisms. We hadn't even considered looking at microbes. So, so what's spiffy about this is you have beetles coming here to feed on the dung, you have flies, and it's not just one kind of beetle, it's not just dung beetles. We have rove beetles, we have pleasing fungus beetles. There's a ton of different kinds of beetles, as well as several different kinds of flies. But what's poop? It's waste, right? It's leftovers. Isn't that weird that you've got all these critters coming in to just feed on leftovers? Well, and even more crazy is the fact that dung beetles actually radiated into this niche and have really expanded through it. We've got over 5,000 species of dung beetles so far identified. And of course, as you well know, there's a number, the majority of, 25% of the macro organisms in the world are insects, most of those being beetles. And again, the dung beetles are a big portion of that. So they're found on every continent except for Antarctica, which is fine. Maryland winters are cold enough for me. I don't want to go out there or go up there. But you can see these different, the green radiation is from Europe and Asia, and then we have specific beetles that radiate on African ungulate dung specifically. And so there's some that are found just in Africa. I can't wait to go find the big elephant dung beetle that's evidently about this big. And then we have some specific to the Americas. So today, this is where I guess I should start using a pointer. Today we're going to be talking more about Onthophagus taurus, the bull-headed dung beetle. And it originally evolved in the Mediterranean, but it's globally distributed since then. So maybe one way that these organisms can feed on dung, this nutrient-poor resource, is through symbiosis. Now this term was originally coined by DeBerry in the 1800s, and it meant simply the living together of different organisms. He did not say, symbiosis is mutualism or parasitism. It's just two totally different things getting together. And when the two of them are together, they make something different, okay? So symbiosis influences life, right? We wouldn't have eukaryotes if it weren't for an initial coming together of two different microbes. They cause changes in the behavior, the physiology, the genome, the ecology. Pretty much every aspect of an organism, both the host and the microbe, are influenced when the two of them come together. Okay? So that amazes me. So whether you are talking about mutualism, where we've got two beneficial interactions, we're benefiting the host and benefiting the microbe, or we're talking about parasitism, where only generally the microbe is what's benefiting and the host has a detriment, we're still talking about this interaction. And of course, humans like to box things up, right? A lot of interactions slide along this scale depending on the context. So, but I'm sure you've heard of mutualisms before. Coral zooxanthellae are one good example of this. So there's a dinoflagellate, a little algae kind of that lives inside of corals that provide the majority of carbon for the corals. And then we have fungus coming together with our algae to make lichens. Well, the symbioses I got obsessed with were insect microbe interactions. So these are really spiffy, and we've had an explosion in people studying these right in the last decade, which has been fabulous. But really the system that was first kind of, we knew a lot about in the 90s was the aphid Buchnera system. And this is where we have these aphids that feed on plant sap. Now plant sap is pretty much Kool-Aid. Okay, can you actually operate well all the time on Kool-Aid and pure sugars? My kids want to, but no, you can't. Neither can aphids, but they have these really cool microbial partners. Buchnera is the majority of them that actually can take those sugars, change them up, and then feed the aphids. And now the aphid can actually do everything it needs to. It can feed, it can reproduce, and as you see here, we even have aphids will reproduce so quickly that we have mothers and daughters, grandmothers and grand aphids all together on the same plant a lot of the time. So our microbes can supplement diet. Tsetse fly is another example of this, where it's feeding on blood. The olive fly is what I worked with during my PhD, and there's many, many other systems. We've also found recently, and so people used to think it was just nutrients, but we've also found recently that there's other microbial interactions that seem to benefit the host. One is temperature fluctuations. So some of the aphids out in Arizona, where it gets really hot, seem to have these secondary symbionts that have come along and helped them survive heat shock. So that's pretty spiffy. We also have natural enemies resistance, my favorite example being the bee wolf. So this little critter, mom smears actinomycete bacteria on the pupa to protect it from fungus while it's pupating. But there's also a parasitoid wasp that attacks aphids, and we found bacteria that can kind of protect the aphids. So there's lots of cool things on the insect side, and people like to focus on the host. But what about poop, right? This is the waste product of another animal. So it really is kind of a nutrient-poor diet, kind of like plant sap in some ways. Herbivore dung is really what, I think I've broken this somehow. So herbivore dung has 86 to 90% of it is undigested cellulose. So you can see lots of bits of grass in there. And additionally, not only does it have the leftovers that your cow has eaten, which think about a cow, right? 4 stomachs, they chew their cud, so they eat it and they throw it back up and they eat it again. They've got 4 stomachs. They've got all these bacteria and flagellates and ciliates helping them, and yet still there's all this grass left in the poop. It's crazy. So Also in that dung, there's going to be a bunch of microbes sloughed off out of the host gut, and there'll be environmental bacteria that are coming in to use this resource. So a lot of times we focus on the host, but I kind of wanted to look more at the microbe, because microbes are cool, right?
They're the best.
Yes. Okay. So I wanted to look at how hosts influence the microbes and the microbial communities, because a lot of times what some people were seeing was that the bacterial genomes would be really reduced in these insects that were feeding on plant saps and that were feeding on blood and things like this, and weevils that were feeding just on corn. And immediately I kind of looked at it and I was like, that's kind of weird, because these things feed on the same diet throughout their life. A lot of them were, or several of them were also hemimetabolous insects, so they look just the same when they're babies as adults, right? So something like an aphid, the only difference in that mom and aphid that I showed you earlier are their body sizes, but their morphology is not changing, okay? So I was excited to look at insects where there's a diet change, so where the larva feeds on one thing and the adult feeds on something else, where the physiology is different. So with whole metabolous insects, like flies and beetles, it's really cool. You've got this maggoty kind of thing, and it has chewing mouth parts, and it eats on plant leaves and stems and flowers and such. And then it throws everything up, and it kind of pupates. So it hunkers down. It starts degrading the majority of its tissues and cells, and then rebuilding those to become this beautiful winged adult that often is a fluid feeder. So with flies, they often have little spongy mouth parts, or they might have a poking mouth part, or beetles have a long tongue. But either way, they're very different from when they were a larva. I also was interested in finding a host, an insect host, where the transmission of the bacteria to the offspring was a little different. In some of the systems that we had previously studied, really the bacteria go into the embryo, and then so they're vertically transmitted inside the embryo. I was more interested in some of these where the bacteria are smeared on the outside, so they have a little bit of an environmental transmission aspect of it, which is, style, so that was a little different too. So what might happen when I find this insect? Well, I found two of these insects, and that's what this was with the, again, with my dissertation work with the olive fly. But I'm just going to tell you about the dung beetles that we're playing with today at Townsend. So things that I thought might change as we have all these changes to the insect they're living inside of. That maybe the taxonomic diversity changes. So maybe they have totally different bacteria. Maybe when we go through pupation, we get rid of a whole bunch of bacteria and we pick up something new. Oh. Maybe community abundance changes. Maybe it's something like what we see in humans, where when babies are fed on breast milk with those sugars that are really different from anything else in the world, we have a totally different ecosystem than what we have in adults when we're eating meat or when we're eating vegetables or whatever. So maybe the community abundance is shifting. They're always there, but changing in how many there are. Maybe there's a change in functional diversity. So maybe we've got the same critters, but they're doing different things, depending on what the diet is and what the structure is they're living inside of. And maybe, finally, maybe there's something different with their genomes. So, poop's been a problem for a long time, especially been a problem with cattle introduction. Cattle are originally from the Mediterranean, and when they've been introduced, of course, all over the world, except for Antarctica. And they produce a lot of poop, okay? So 65 pounds, that's like, the equivalent of, gosh, I guess my fifth grader, or fourth grader. Yeah, I'm not graduating yet. So that's a lot of poop in one day, in one animal. And if you don't have beetles, that poop's going to pile up on the environment. So this is a shot from Australia when they first brought in cattle. Here in the highlands in China, this particular group of people actually they pick up the cow pats and they use them to build houses. They use them for food. So there's many different ways to deal with this dung, but dung beetles are a natural solution. So the bull-headed dung beetle, strangely enough, evolved in the Mediterranean with the bull-headed cow, and it was introduced into the United States and Australia. So in the US it was introduced accidentally. In Australia, when they brought so many cattle and had so much trouble with the dung problem because the native dung beetles knew how to deal with kangaroo poo, but they didn't know they couldn't handle, maybe because they didn't have the right microbes, they couldn't handle cow dung, so they would leave it alone and it would pile up. So Australia actually, the Australian government went out and collected beetles from all over the world, brought them in to try and see if they could establish some dung beetles that could deal with the poop problem. So they're economically important because we need to move that dung below the surface. This is part of the cycling of nutrients in our ecosystem, right? So this isn't just, hey, it looks gross, but we need to remove habitat for pest flies. This is a big problem in Australia again. You get bush flies that grow in the dung. It returns nitrogen to the soil so that our plants can grow. And also, something that bothers me around here especially is, if your dung's on the soil, on the top of the soil, it can run off into your natural waterways. So that's a big problem. What's really cool about these dung beetles is they completely degrade the dung. The larvae, they have this little niche partitioning because holometallous insects, right? They're cool. Larvae feed on the solid dung, and so they're chomping away at the cellulose bits. And the adult has a sieve-like mouth part where it kind of sieves through and feeds on the liquid parts. So I'm going to tell you more about Onthophagus taurus, the tunneling dung beetle. It's a really crazy beetle, okay? It's neat in that, the mom says, one day, son, this will all be yours, because she really puts a lot of effort into making this brood ball for her baby, for her egg. The offspring stays in this same brood ball the entire part of its larval stage, okay? So this is, of course, staged with several different brood balls, but you can see how they're at different stages, and the brood ball, the brood chamber, starts getting eaten out as the larva gets bigger and bigger. So it's a safe environment, it's a safe house for mom's egg. We know she provisions with food because that brood ball gets smaller over time, but maybe she provisions with something else. Maybe microbes. So this was what I was excited to look at, is to see if these dung beetles had vertically transmitted endosymbionts that could allow them to feed on dung. So a few expectations. If there was a specific microbiome, we would expect that it would be something that they'd get from their moms, okay? In the case of Taurus, only the mom makes the brood ball, so that kind of simplifies things a little bit. I would expect that the microbes would be completely distinct from what we'd find in the dung. We'd find similar microbiomes in the adult and the offspring. We'd find similar microbiomes across different populations if they're really highly co-evolved, and they'd be important to the beetle's life cycle. So I started attacking this first question years ago at Towson, actually as a visiting assistant professor, because we don't have olive trees here in Maryland. So I started working on the dung beetles instead. And we started by collaborating with some colleagues in Indiana University to do this sterile rearing experiment. And what I wanted to do here was to see, is the microbial taxa that's in the mom the same as what's in her offspring? We would expect that. We would also expect a similar microbiome perhaps in adults and a closed offspring. So if it's not transmitted by the brood ball, we would see something different between the mom and her offspring. It would either be there would be no microbiome, which we have that in some systems where the baby's just born without and then dies usually pretty quickly, or we have a perturbed microbiome. So maybe it looks like human skin microbes, or maybe it looks like the dung or something like that. So our colleagues at Indiana went to North Carolina and collected a whole lot of female dung beetles from the wild. They brought them into the lab and they provided them autoclaved dung, autoclaved dirt, and water. And you can see that each one, well, hopefully you can. So this is a brood box where they would put each mom, and one mom would stay in this brood box, and she would make several little tunnels and brood balls. And some, of course, didn't make any, but they would make brood balls weekly. Those brood balls would be taken out so we could stage them and know where they were in development. And they were sacrificed at different stages. So first we had to make sure we weren't introducing any microbes to the system. So again, the soil and the dung were autoclaved. The dung was stored frozen. Additionally, we tried to culture bacterial colonies. We couldn't get anything to culture. But then, of course, sometimes you can't get bacteria to culture. So we did PCR, and we specifically made sure that there were no PCR inhibitors, and we still amplified nothing. So it seemed like our protocol for sterilizing the dung and the soil was successful. We wanted to make sure that we were looking at something that lives inside the beetle. We don't care about something that's on the outside. Sometimes that's helpful, but we really wanted to see, are these bacteria inside the offspring? And so this is where it's nice to work with insects and beetles instead of humans. You can't put humans in little test tubes and vortex them about, but you can with beetles. So I cooled them down so that they would be kind of asleep, and then put them in a mix of bleach and detergent. They've got all these ridges and such and hairs on their bodies, so we need to have that detergent in there to make sure that we can actually get this solution of bleach and everything to the insect exoskeleton and wash that off. And then we rinsed it with sterile water, plated the rinsed water, extracted DNA from it as well. Again, we couldn't amplify any 16S rRNA or culture anything, so it seemed like we were successfully sterilizing the outside of our beetles.
It didn't kill them?
Nope. And then they would get up and they'd kind of stagger around a little bit first, but they're fine. They're tanks. The olive flies can handle this too, and they're the same way. It's crazy. So this is just showing you what the data look like here with, these are different surface sterilizations. And this is Carla Fieser, who was one of my very first students at Towson who helped with this project. You can see this is our plate that has nothing on it, so you shouldn't see. So when I was a visiting assistant professor, we were kind of limited in funds. And so all we could do really is these, just a few cloning reactions. So we took 2 of the females out of, and 17 of the offspring, and we extracted the DNA and then tried to amplify pretty much the entire 16S of the bacteria. So when you do this, though, you have to separate out all the different, you can get a whole lot of 16S samples, and so you need to separate them out. So we did that with cloning, and we only picked 10 to 15 clones per sample because that's how much money we had at the time. And we produced a chart that I want to explain to you because you'll see more of them. This is a heat map. So on this, the y-axis is the family of the bacteria that we had found. We have on the x-axis are our samples. The 2 red boxed samples are our female parents, and the rest are the offspring. This clustering indicates what has more similar microbiome communities. And here is our abundance scale. So if there's yellow, it actually means there's nothing there. If it's dark red, it's at least 90% of that one taxa. So you can see that our taxa really kind of, even with these few clones that we had, they separated out to where we had one clade that really had Enterobacteriaceae, and the other had a mix of Commonwealeridaceae and Pseudomoneridaceae and Enterobacteriaceae. So what was spiffy is the females were on one side of this cluster, and the majority of larvae were on the other, though we do have some larvae mixed in here as well as pupa. So that was kind of interesting. We saw two different microbiome types, one that seemed associated more with the females. So I had an REU fellow come down from Ursinus College that next summer, and she actually, we had isolated these bacteria, so she went in and used two other housekeeping genes, LEP-A and JAR-B, to further identify what these bacteria are, because 16S sometimes only gets you so far. And so these were the bacteria that we found that we can culture, we've got in the lab, and it's where what my students are working on now. So when we did that initial experiment, the sterile rearing experiment, our colleagues actually had a number of females. They had 17 female parents, they had 101 offspring, and so when I got to my postdoc at IGS, my very generous postdoc advisor said, sure, we'll sequence that, no biggie. So I extracted all the DNA and then sequenced these using a next generation sequencer. So now instead of getting 10 to 15 reads per sample, I'm getting 23,000. Kind of makes a difference, you'd think, right? But not really, in that we found a lot of the same taxa. We're still finding Discomonas, Commonwealeridaceae, Pseudomonas. So that was kind of interesting. Here, so this is the same abundance key where it's going from yellow, which is nothing, to red, which is highly abundant. And then here is the life stage. So our red and our yellow are adults. The red is the female parent. The yellow is the closed adult, so her offspring, but as an adult. The green are our larvae, and then the blue are pupa. So we found similar taxa to what we found with Sanger sequencing with just the 10 to 15 clones, which was surprising but really exciting too. And the adults and offspring do seem to have similar taxa when they're in this adult stage morphology, but they may have a different abundance as well. Okay, so back to our big question, is this a specific microbiome? Is it acquired from the female parent? We think it would be. How would that happen? So this is where we went back and looked at the brood ball again. So again, this is from our colleagues in Indiana. They sent some brood balls down so that I could do some electron microscopy on them. So I compared the brood ball chamber, this little safe home that the mom puts her egg in, and then the area, so here's the egg with the brood ball chamber, and then the back part of the brood ball, is actually just a solid, it's more solid with more grassy kind of structures in it, and then a fecal pedestal, and I'll show you what that means in a minute. So if the microbiome's coming from the mom, we're expecting that they'll be transmitted via these chamber structures. Because the brood ball chamber looks different from the rest of the brood ball, we have a pedestal that the egg is sitting on. But if it's transmitted throughout the entire brood ball, we would see no difference in the microbes and the structure between the dung in the chamber and the rest of the brood ball. So the larva, or the egg, is actually laid on this little pedestal, and this is what it looks like under electron microscopy. So it has different structure. Here's the brood ball chamber that's running in this direction, and the pedestal's just pooped again on top. So it's a different structure. That was kind of interesting. What happens is when the larva comes out of that egg, it feeds on the fecal pedestal first. Then it feeds on the brood ball wall. So I looked in the pedestal, and I did seem to find some structures that looked kind of like bacteria, but it's electron microscopy. You really can't do much more than, hey, it looks like a bacteria. But you know, it was this initial transmission story that had captivated me long ago as a PhD student when a developmental biologist said, hey, you like insects, let me tell you this story. And his story was that this dung beetle, so the mom makes this capsule, she has a pedestal, the larva feeds on the pedestal, and then it feeds up one side of the brood ball wall, turns around and eats its own poop as it's coming back down, and then it goes up and down feeding more. So when I heard this, as someone who's excited about microbes, I went, oh my goodness, this is a concentrating mechanism. This is a way that these insects are actually pulling in microbes from mom, and then whatever microbes are helping them digest that brood ball that they're on, they're growing up in their gut, and they're eating as they come back down to make those grow more. So that's why I was so excited about this system in the first place and when I started working with it. So if you look at back to electron microscopy in the chamber, you can see the chamber actually has a smooth matrix, whereas the back part of the brood ball is more cellulosic. When we scraped underneath that matrix, we did see bacterial-like structures. They're pretty long, but one of the bacteria that we found is the Lysnobacillus, which can have pretty long structures. So again, it's EM. It's hard to know, but it kind of at least gave us a little bit more of an idea of what might be going on. So it looked like the bacteria are being provided through the brood ball. But there was still this question. So this is all with the sterile rearing work, right? But people kept saying to me, yeah, okay, so you've autoclaved dung, they've grown up on dung, but in the wild, don't they probably pick up the bacteria from the dung? And that would make sense, right? They're feeding on it. In fact, a lot of the dung beetle natural history is that what these beetles are sieving through and eating in the dung is the bacteria and the fungus. So I wanted to compare the beetles and the dung microbiome in the field. I've told you about the sieve-like mouth parts already. And so what we did is we went out to dung pats, and we would take and we would collect an animal and then collect another one right beside it. And one of them we would immediately kill in ethanol or in acetone, and the other one we would hold on sterile soil for anywhere from 12 to 48 hours. So the idea here was that if they're not getting anything more in, if they're actually, they'll poop out any bacteria that they've gotten from the dung, and so what's left in their gut should just be their native microbiome. So I expected that if the bacteria were acquired from the dung, we'd see the same thing in these fed and these starved beetles. Okay, it would be no different. And of course the dung microbiome as well, we sampled the dung too. But if it comes from the female, then we might see that the fed beetle microbiome is similar to the dung, but starved beetle microbiome would be different. So we looked at that, and again, another heat map. This time, the beetles are all in red, so you can, the beetles form this distinct clade. I'm not sure why they clustered within that clade, but this is what we have so far. And then it didn't matter what kind of dung even that we collected off of. These are beetles that are collected off of horse dung, that's the purple, goat dung is the blue, dairy cow dung is green, and the yellow is beef cow dung, and yes, beef and dairy cow dung are very different. I've learned so much about scat. But either way, you've got this really distinct clade where you've got a grouping just of your dung microbiome, and you have a grouping just of your beetle microbiome, and that really blew my mind. I didn't think it would be so distinct, quite honestly. They do have a few shared taxa, but they're really low abundance, so less than 5 reads out of 23,000, which is probably more of a sampling issue, more of a taxa filtering issue than anything else. So I decided that the dung beetle, or felt like this data supported the idea that dung beetle has a microbiome distinct from the dung. Since then, another group has done this, and they found the same thing in a different dung beetle species, so that was pretty exciting. what was really surprising, though, is that I did find very similar OTUs between the beetles that were killed immediately and those held on the sterile dung. So this made me think, hey, they're really not picking up anything from the dung, and they're really not, their microbiome just isn't shifting. It's really robust and stable, which was quite a surprise. But again, it supported the idea that we see these different microbiomes with the beetles versus the dung. Okay, so now we see that we've done the sterile rearing. Our offspring have similar microbiome taxa, but different relative abundance. The beetle and dung microbiome are very different. It has a very stable microbiome. So what about the adults and the offspring? Well, we would expect that if the microbiome is essential for the beetle, we'd see the same taxa all the time, right? But if it's not essential, maybe we'll have different taxa present in different life stages or individuals. This is at least where I started thinking. So this is again that MiSeq sequencing where we had the 23,000 reads per sample. And I've already talked through this a little bit in that I'm going to show you the adult, anything with the adult body plan clusters together, and then those with the larva, these are larva and pupa, and then these are younger larvae with younger larvae over here. One exception being these samples where we do have these two yellow samples that are the adult recently enclosed animals that look a little funny. So we need to look into that more. But overall, it seemed like we really had changes in relative abundance and shifts in the taxa across life stages. So there was one thing in the larva, and there was something totally different in the adult stage. So I wondered if this was because of the diet. Remember, there's more cellulose with the larva than the juveniles. But once I saw these data that have the unfed adult offspring, that didn't make any sense. So now maybe it's really that it's the digestive system morphology or maybe it's the hormones or something else, something about the physiology that's changing. So this is where we're kind of headed now. This is one of our directions in the lab, is looking to see if it's diet or gut morphology, selecting for the different taxa. And coming to Towson as a assistant professor now and reestablishing my lab, we're going to start by looking at a lot of the culturable microbes. So I want to take the isolates that we already have and we want to grow them on dung. We want to see if they degrade cellulose, because that's one of our hypotheses, right? We want to see if they harbor antibiotic resistance. You've probably heard a lot about antibiotic resistance and its use and the use of antibiotics in agriculture. This is an issue. So we wanted to see maybe our dung beetles are getting exposed to levels of antibiotics. They're harboring antibiotic resistance and then a vector for that. And we also want to see if what these cultures have as their metabolic phenotype. So some of this was done before as a visiting assistant professor. Lottie was a fabulous student who did a lot of the initial cloning and dissection and isolated these bacteria that we are focused on now. And she did, in one of her initial experiments, see different densities of bacteria and different types of bacteria in the midgut versus the hindgut. And this is something my students are going to work again on, well, my students and I are going to be doing this summer. So at IGS, as a postdoc, I was lucky enough to work with Ms. Kenyane King from Western High School. She's now at UMBC in her junior year, so she was awesome to work with and is just a brilliant person. So but probably a little bit shocked on her first day when she was only in the lab for 6 weeks, and the first thing I hand her is a mixer and a bag of frozen dung and said, let's make dung agar plates. So she tried to grow these cultures and see, would they grow on dung, so this is her special dung agar mixture, or would they grow just on agar? And she didn't get any growth on the agar. This is where she pressed a little bit hard with this. But she did see growth with all 4 different culture isolates we were focused on the dung. So that was pretty cool. So some of these bacterial isolates seem like they can degrade dung. Now this is chopped up dung, so that's another thing that's kind of interesting. Then we wanted to look at cellulose. And so she had 1 week left and very bravely spotted isolates onto this LB with 1% cellulose methylcellulose. I can't do that right now. CMC plates to see if these bacteria actually are secreting cellulases. And this is a collaboration with Jeffrey Gardner down at UMBC. So he gave us a controlled bacillus, which is an extreme secretor, is how he classifies it. And then, and I realize this is just 1 plate, and this is the beginning of this, but it looked like our Providencia and our Pedobacter both seemed to secrete a little bit of the cellulases. So this is, again, something else that a different student's going to be working on this summer. So it gave us hope that maybe some members of the microbiome are degrading cellulose. The other thing we're doing is to look at the microbial phenotype effectively. So what substrates, what can these cultures feed on? And so this is part of what a lot of what a startup went by. And we just got it 2 weeks ago, so no data yet, but this is what is to come in the summer. This is Ms. Allison Moss, one of my students, who is proudly showing off our BioLog microstation. We have Gen 3 plates, which is a way to look and see what kinds of different sugars can these microbes feed on? What kind of pH do they live on? Can they feed on lactic acid? Can they feed on, what amino acids can they feed on? So this is a way for us to get a metabolic phenotype, a fingerprint effectively, of these microbial cultures using all these different reactions in 1 day. And we're also going to use these, so the Gen 3 system we're using just with our specific isolates, but they also have eco plates where you can actually go out into the environment, like the only environment I care about, beetle guts, and you can plate that on your plate and see how that environment of those environmental microbes actually respond. So that's some of our summer work. Back to Kenyane's work, another thing she did was to see if our microbes had antibiotic resistance. Antibiotics are used on farms both preventatively and for treatment of infections, mastitis being the biggest problem with dairy cows. We tested 4 different antibiotics because they have different ways to, different mechanisms of action. So streptomycin and oxytetracycline inhibit protein synthesis, whereas ampicillin and C4 are disrupting cell wall synthesis. And they're both commonly used in the dairy industry. So she plated our cultures on Mueller-Hinton media, and then what we would expect is if the bacteria are resistant, you're not going to see anything growing because they're going to die. But if they're sensitive, then the bacteria around where you put that antibiotic disc are going to die off. Okay? So this is what she saw. Kenyane found that every one of our isolates was resistant to some kind of antibiotic commonly used on the dairy farm. Now, does that say definitively, hey, these are coming from due to exposure in the dairy industry? No, it just, because antibiotics are also natural also found in nature. So we don't know where this resistance is coming from. Maybe it's native to them, maybe it's something they're picking up, but it's just something to think about. And the student who's going to continue with this work is Sarah Smith. She's going to screen additional taxa as well as additional antibiotics. She has quite the spreadsheet of antibiotics she's going to look through. And then we have a common garden experiment that a colleague in Minnesota ran where they exposed beetles to antibiotics in the dung or not, and we're going to see how that changed their microbiome. So other future directions, we really want to look, we've kind of been looking a little bit at 16S, looking at what the bacterial diversity and abundance looks like. I'd like to also do this with metagenomics, and that's what we can do with our Common Garden experiment. We'd love to get to the transcriptome stage, but more likely we're going to do genomics next, because we can still partner with Institute for Genome Sciences, get these bacterial genomes sequenced, and then hopefully either students in my lab or in the classroom can annotate those genomes and see what's in them. And again, all of this is so that we can determine what are these microbes doing and how are they interacting with their host. So is there a specific microbiome of dung beetles? Seems like it. It also seems important for beetle life history, and that's some more data that I don't have time to go into. But it's another one of those places we're looking forward to in several years, where we can grow up our isolates, have a dung beetle colony, and actually introduce the bacteria at different times and see how that changes the beetle activity. So it does seem like these microbes are dung and cellulose degrading. Some of them have antibiotic resistance, and this is where we'd like to go to, is with some of this larval development work. We do have collaborators at Indiana who have done some initial tests that seem interesting, but we want to follow up. So I would like to thank you for your time and for your attention. But I'd also like to thank all the different people that have helped through this. So at Towson, I've had several different fabulous students, and I'm really excited to start the lab back up with these two extremely excited students. They both emailed me before I even started a position at Towson, which was overwhelming but exciting. These are collaborators at Indiana University, Institute for Genome Sciences, Salisbury University. One of the projects I didn't have time to tell you about was a project that I had with Mallory Hagedorn at Salisbury University and Dana Price. Dana is an amazing dung beetle taxonomist, and Mallory is an amazing student, and she got an NSF grant to, graduate school grant to look at dung beetle microbiomes across Maryland. So that was another fun thing. I also didn't get to tell you about my work in South Australia with those dung beetles, so that's coming out soon. And then also Jeffrey Gardner, who collaborates with the cellulase work. And we've had funding from several different places and scrapped together a lot of this initial work. But I'm also very, very proud of Allison, who just got the Garden Club of America award. So she'll have a little bit of funding for her this summer to continue some of this work. So thank you so very much for this opportunity, and thanks again for your attention. I'd love questions.